Sounds better to me. Is that better, chat?
Let me know if that is better. Yeah,
Perfect.
much better. Okay, good. Good.
Awesome. Danny,
I
how?
just gotta remember to turn it back down when I start streaming. But yeah, I'm doing well. Yeah, happy Bat Appreciation Day, Valent.
You too. How was the stream? What was the? I guess what was your favorite bat highlight?
Oh boy. I think being able to. Oh, we watched a neat little clip about bats in my part of the world, near the San Francisco Bay Area. And there's a bridge near Sacramento, outside of the Bay Area, but there's thousands upon thousands of bats that roost under this bridge, and that was really really cool to see. And especially when we learned that they actually engineered the bridge to provide a living space for the bats. There's like a little recess underneath the bridge, and there's a piece of wood that runs down it lengthwise, and so the bats have got a nice little spot. It's just bat size on either side of this long piece of wood. Um, and it's just a perfect spot for them. So
That's
that was pretty cool that people are thinking about the bats as they're building bridges.
yeah, it's really neat. So is it like a almost like a cheaper version of a bat box?
I guess so, except it's 7,000 times longer or whatever.
For sure.
So,
Like I guess not
yeah.
not encased, but a place for them to roost.
Yeah.
Nice.
Yeah.
You were mentioning on stream too that you're gonna be putting up your bat box soon.
Yeah, still gotta find a good spot for it where it can get sunlight during most of the day to maintain a nice high temperature for the bats, but also enough space underneath where they can drop down and build up enough speed that they can spread their wings and take off. Because most bats have difficulty taking off from the ground, so you gotta put the bat box way up high to give them enough space to drop. Yeah.
That's really cool. Do you have a favorite bat, Danny?
It would have to be a fossil bat. Yeah, like, what was it? Icaro nectar? I forget I'm not great with bat names. But there's these two bat genera from the Green River Formation of Wyoming. They're actually from not too far from where I'm going to be digging this summer.
Oh, cool.
In a little over a month, so yeah. The fossil history of bats is it's both really good and really poor. Like, we've got exquisite fossils of bats, but we don't have any, like, proto-bats yet. So they just kind of show up in the fossil record almost fully formed. And someday somebody's going to find some proto-bats, but it hasn't happened yet.
What do you imagine a proto-bat to look like? Like, as in, is it a longer wing set that spans from, like, foot to arm, like, kind of like a flying squirrel, or, like, what are you envisioning?
Probably a lot like a colugo, if you know what that is. They're sometimes called, like, gliding lemurs.
Yeah.
They're related to primates. Yeah. We talked about them a bit, too. They're super cool. I would imagine it would be kind of like that, but a little mousier looking, maybe.
Okay.
Yeah.
That'll be really cool to find. I wonder why it hasn't been. I mean, it might just be like you always tell us, like, time, location, preservation, and people where they've dug, right? That maybe it's not enough depth to or investment of money looking into it, but
it seems to me we just don't have a good, like, Lagerstätten fossil site, like a site with exquisite fossil preservation from that period of time yet. We've got some from the Eocene, from like 10 million years after the extinction of most dinosaurs. But we don't have anything from just after the extinction event yet, and that's probably when the first bats evolved. Was probably just after. There's this big, like, vacuum that opens up that with the extinction of the dinosaurs, mammals can just kind of go crazy diverse all of a sudden. That's probably when the first bats evolved, and we just don't have the right fossil deposits from that time.
Gotcha. It's frustrating because, I mean, it's the same thing with some insect fossils too. It's just like it's not the right scenario or not the right setting for you to get answers. And like we talked about with the Helland too. Like, there's no real, everything that we have is just a guess in terms of behavior and social structure. Like, yeah, we have the exoskeleton, but there you just haven't been able to find the actual fossil that you need to be able to do the information that we were trying to get out of it.
there was something we were talking about today too that we wanted to get your take on, Bullet. When we were talking about bats are reservoirs for all kinds of different diseases, and bats can be just chock full of disease without necessarily any harm befalling them, because they've got this really interesting immune system where they've got super high metabolism, they've got really high body temperature, they're very, very active animals, and yet, like all of these different conditions that you would think would like increase the rates of cancer or other diseases, instead they have apparently very, very low rates of cancer, and their immune system doesn't overreact to things. And also, it seems like they don't have a lot of like DNA degradation that would come with high temperature, high metabolism. They seem to have like, we were watching a video about, it's hypothesized they've got some sort of specialty DNA repair thing going on. And we thought we should ask you about that and if that would make sense.
So that's actually ties into a little bit of one of the papers we're talking about today, right?
With tardigrades, right?
Yeah. And so there has been some sequencing done on bats to figure out, right, what's different. And you mentioned even some RNA sequencing that shows there's higher metabolic activity and there's higher immune activity. It does seem like there are some, like a unique population of DNA repair genes. So it's not just that there's more of them, there's like this additional like mutation that a lot of species have that seems to be active that isn't present in humans. It's the analogy that I've heard. It's similar to how I know we keep going back to ants, but it's in some queen ants. There are actually this additional DNA machinery that's turned on that's not turned on in other workers, and so that's why those are suspected to be able to live longer. It's because there's this species of RNA or this gene that's turned on. And interestingly enough, with bats, that social signal seems to be very important to turn those things on. So if you take them out of that social setting, those genes turn back down. So there's this like feedback loop that's going together.
Interesting.
So it's which is why, like, we had a collaborator who was trying to do proteomics on them and figuring out what proteins are, because he thought that this was epigenetic, right? That these are switches that are turned on and off to make things more active or less active. And it turned out that the lab he was collaborating with, the way they kept their bats, I guess, was stressful for them, because, and it wasn't known at the time, like people didn't know, but that they were not, like, they were in groups, but not the groups in terms of the numbers there were in the wild.
Sure.
And that was negatively affecting the fitness, at least the hypothesis of the baseline that they were having. So it's a super complicated species, like all kinds of bats, especially with anything with the social element to it. Like last week we talked a paper about hyenas, and that their social status is epigenetically marked in their offspring. And
wild.
Right, it's just wild. And the way they knew it was they went in and actually tested fecal matter, because they looked at wild populations, because the argument was we can't look at like captive populations because it's just like not the real state. Like it's such a contrived setting that we have that you have to test it in a wild population. You're trying to figure out how you can test them, and they actually found like there's immune genes there too that are differentially regulated. They're higher versus lower tier, lower social setting animals.
Right.
And Charlie's Dragons is right. The higher metabolic rate, so higher metabolism, usually leads to more species of superoxide dismutase, which is two oxygens bound together with a negative charge that has an affinity for DNA and hitting DNA, which can cause mutations. And that's why you eat antioxidants, right? So those are the oxidants. You eat antioxidants to reverse your aging. And there are an absurd number of studies that show if you starve an animal, like you limit caloric intake to slow metabolism, it extends lifespan. Whether that be in insects, it's been done in mammals.
Lab mice.
Yeah, fish. Like, even budding yeast, Danny.
Really?
Like it seems like it's a core feature of organisms.
Of life on Earth.
Yeah.
Yeah, yeah.
And yet here we are, like, you know, if you put a pizza in front of me, I could probably demolish the pizza.
Me too. Oh yeah, yeah. Oh boy.
So it's I mean, everything in life is a balance, but it really goes to show, like, our Toxicology 101 class where folks said, you know, the first thing that we learned about in that class was everything at some concentration that is a toxin. Like, everything at some concentration can kill you, and it's kind of weird to think about, like, food is almost that, and not like in a way of a weight gain, but so much as like the side effect of metabolism can induce DNA damage, which is, I don't know, it's kind of wild to think about that. That's how life works.
It makes sense though when you stop and think about it, but it's on its face it does seem kind of crazy.
Oh yeah, totally makes sense. Like the biology behind it makes sense. It's just like, really?
Yeah.
Come on, I just like I really like to eat.
Yep. Yeah. Oh man.
And that's actually like there's been studies done. You know, when like an Olympian comes into the ER, and unfortunately we lose them. That seems to be tied with metabolism, which also is tied to workout. So because when you're working out really, really hard, if it's not balanced right with working with a nutritionist and other kind of doctors, you can have an increased level of this metabolism, and therefore the superoxide dismutase, and it can yield to a lot of disease states, yield like a heart attack even, because it can be damaging heart cells. And so it's which is again really counterintuitive at face value of exercise being almost bad in certain doses. But again, it just comes back to things in moderation.
Makes a lot of sense. Yeah, yeah. Hey, would that be a nice segue into the tardigrade paper?
Yeah, if you don't mind. I thought it was a really interesting paper, and I apologize, Danny. I didn't realize that the one that I sent you was behind a paywall.
Uh-huh.
Because I was on this computer and I still have my sneaky access.
Yeah.
And so, y'all, I unfortunately sent Danny a paywalled paper, and then when I
luckily there was a press release that went with it, which was nice and explanatory.
So I actually went and instead of trying to hack the system, I found this. They actually posted a preprint of it on BioArchive,
nice.
And I just put it in the chat for y'all.
Okay.
So this is like think of it like an early release of the paper, chat, where instead of going along with publication, you upload it to this database. Essentially, it's free. This is like the only no charge location for a paper. There is a basic mechanism for folks to look at and quality control. I know someone who does this, and all they basically look at is this a joke or not? Like, that's the quality control that we have. And then they post it on live, and actually there's a really nice interface where you can submit your paper from this website to journals that accept preprints. The really big tier journals still do not accept preprints because it takes away their sheen and their media embargo. Like, people will go to this website instead of their own, and they don't get the attention. It's frustrating, right? But that's unfortunately how it works.
Yeah.
But I did find the BioArchive equivalent. I was like, okay, this is what I shared on our Discord too, of like for folks to check out, because it's free. You're not having to use any kind of different means to go through and get the paper, and it's the same stuff. And Danny, I'm so sorry, just it auto loaded like with the creds.
No
Didn't even realize that it was behind a paywall until it was too late this afternoon.
What I should have done was when I was at the university library yesterday, I should have looked and downloaded it then, but it was not top of mind.
No, I try to be really aware of that, and I'm sorry that I totally missed that it was behind a paywall.
No worries. Hopefully soon I'll have institutional access through that university website at the university library that I can access from home, but not yet. I'm still kind of waiting on that.
Fingers crossed. And Dr. A says, astronomy and physics have archive, and it's just accepted practice to post there. Really? Nice, Dr. A, that it's accepted to do that. See, in bio, for cell science and nature, you do not post it if you want to try to get your paper there. And then my last professor was very she didn't believe in it. She thought that it would mean that people could still steal your work, even though the whole point of it is that it's a timestamp on it, right? It's meant to protect
Yeah,
you.
you could point to it and go, "Hey, I posted it at this time." You've got a record. If anything, it's the opposite. Like you could demonstrate the. It's like submitting an abstract at a conference. You know, you're proving that you have that idea and you're getting it out there at a certain time.
I guess there's been a few, not, I don't know, maybe horror stories of people surfing bioarchive for easy, hard-hitting experiments, and they'll do like figure one, so like the initial observation, and publish it in a not top of the line journal, but like one of those intermediates, and then when the top of the line gets it, and you have it written like, this is the first blah 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 of this, and they're like, no, it's not. We just saw the press release in this middle tier journal of this observation. And
we should have some sort of tribunal for that, you know, where we'll have to. They'll be forced to wear an embarrassing hat at their next conference or something like that.
And Geeky Heart, thank you for that gifted to Paleo Ken. Ken, enjoy that ad-free viewing and the emotes. And
nice.
Hi, Darkus. And also, it's a piece of raw science for you, Ken. A quick aside, Danny. Ken came in yesterday and was looking at old YouTube videos that you had, and he was like, I didn't realize that your all's friendship like extended back into like 2001. When you were, because we weren't streaming yet, but we were always in your stream asking questions and learning. And Ken was like, "Oh, that's really cool." Like he's like, "I didn't realize that y'all knew each other for like so many years now."
Since 2020 or 2021, something like that.
Yeah. And I was like, "Yeah, we go." I'm like, the reason we're on Twitch is because of Danny. It's like we go way back. 2021. Yeah, thank you, Claire. Or maybe it was even 2020. It was probably that early. But yeah, with the paper story, my postdoc mentor, when he was a graduate student, they submitted a paper to Nature, and it was describing this phenomenon, fruit flies, and the reviews took a year, and it ended in a rejection. And the reviewers are anonymous, and 3 months later, the exact same paper is published in Nature using mice.
By one of the reviewers? Yeah. That happened to a friend of mine too, where, he was like, somebody had calibrated some dates wrong in the Cretaceous in North America, and then that was part of his thesis, was like going through this, correcting this mistake, and he went to publish it, and that guy who made the mistake ended up as a reviewer on the paper, held it up for like a year or two, and then basically published out from under him. He published a new calibration of it to kind of cover his own butt there. Yeah.
Oh.
It's frustrating sometimes.
Yeah. And because he did it in mice too, so it made it an even higher caliber.
Oh boy, yeah.
Because like the impact, quote unquote, right, was this like bigger thing because it was in mice, but all the difficulty was figuring out what genes were interacting, and because things are conserved, you already knew your candidates. You just and if you read the paper, the justification's insane. It's like, oh yeah, based on the literature, we tested, we knew what it was. It was like, yeah, the literature you stole.
That's so scummy. Yeah. That's so frustrating.
But
again, there should be some sort of a tribunal or something, and like there should be sentenced to have to wear a really goofy, embarrassing hat to every conference for the next 5 years or something like that.
Oh man, I would love that. There'd be like some kind of or like mentors that don't appreciate their students or treat them well too, like having the kind of same, like dunce hat or something. But
there are whispered networks, but it's like this sort of thing should be out in the open. Like there should be some outward indication of that bad reputation. But
there's anonymous Twitter accounts that are they're run by. I met her. She's an outed student from academia because of she had a disability, like present tense, and was forced out of academics by a very nasty professor. And so she started this anonymous database to basically be like, what exactly happens, and to also publicly post and ask questions. And yeah, so like some of these people are on that list now, and it's she. The only reason I know her is because I met her at a conference on accident. She's like, "Hey, by the way, this is me." Like I heard, I was like, "Oh, well, yeah, obviously we'll keep it secret," but like, wow, it's frustrating. Some of that happens.
And there's a similar sort of thing with, like my old mentor at Berkeley. She told me that at SVP, the Society of Vertebrate Paleontology, every year, she and some other woman paleontologists would like call aside, female students, and be like, watch out for this guy, watch out for that guy, watch out for this guy, and just have to have a talk like that with, with young women, because there's some problem people out there, and you got to be aware of them and be wary of them.
I actually met with a student yesterday, and she was saying the same. She's like, "Can I talk to you confidentially?" I'm like, "Yeah, what's up?" And she's like, "There is my professor is trying to force me out because I want a kid. What do I do?" And I'm like, well, that's a really hard question. It's a really, really hard question to answer, because what do you want out of life almost comes into play. And then she was like, "I'm already pregnant," and it's like, yep. What's most important to you, and that shouldn't be the issue. It should be open to people.
Absolutely, yeah. I mean, and can she go to HR or something? I know sometimes graduate students are not really covered under that. Universities can be really, really bad about that kind of thing sometimes.
There's no HR protection for graduate students and postdocs. There are some universities now that have unionized.
Yeah, and that's kind of what you got to do. And just about any job, honestly, but especially in a job like that.
Yeah. Oh yeah. Charlie's Dragons, Lita's postdoc contract was ended because she had a kid. It was said, your priorities have changed, was the quote. Right. So yeah, if people tell you that men and women are equal in science, don't believe them. Do not believe them.
It's not true. Yeah. Yeah.
It is a public facing great dream that should be the goal.
We have, but we're not there yet. No.
It is way far away from there. And hi, JD. Welcome in. JD does that. Which is very unfortunate. And this
is true.
The problem is enacting change is hard, because the people that keep going into the system are the ones that were indoctrinated into the system, and it just kind of keeps looping.
Exactly. And also, the people who are more geared toward kind of climbing the hierarchy there are the people who are less likely to try and rock the boat and disrupt the system, because they benefit from how the system works. So, yeah.
On a lighter note, should we talk about some
tardigrades?
Some tardigrades?
Yeah, let's do it.
So, just a little background for those who might not know about tardigrades. So, these are studied a lot because they are survivors. They are arguably extremophiles, that they can survive in, high levels of acidity and high temperatures and high pressures. They can even survive without oxygen. So, you can send these critters.
Space.
Yeah. And there's 1,000 species of these critters. Right, 1,000 of them, and they all have like different kinds of speciality, and people have always wondered.
1,000 so far, by the way. There will be more. They just have not been named yet. I am sure. There could be. I don't know how many species of tardigrade there might be, but it's going to be a lot.
I do wonder, Danny, if any have gone extinct, like how many we've missed as well because of that. Even though they survived so well these extreme conditions.
You mean in the recent era, like how many during human? I'm sure it's been a lot.
Yeah.
Probably specialist ones that only exist in they're endemic to really, really small region or something.
Yeah, so it's like, I guess, even though that they're specialized, they survive so well, they can still be under threat of extinction. So it's not a one-off, we're great at surviving and then we survive everything. It's there is nothing immune from human tampering, I guess is what I was saying.
Exactly, and I would not be shocked. In fact, I'd be surprised if it weren't the case that if any did go extinct, it would be because other species of tardigrades were introduced by humans accidentally or otherwise to their environments, and these super specialized ones from a small island or something just got completely wiped out by ones that were introduced from the mainland or something. That's often what happens with critters like this.
Well, there's even worry that there's going to be contamination of space samples and specimens with tardigrades because they're so good at surviving. Or at least, maybe not the niche ones, but there are a couple of them that are really, really good. And so it's been a long time question of, well, there's all these different stressors. What are the animals using to survive these different stressors? So this study that was kicked off, so it's actually from UNC, Chapel Hill, they're looking at the responses to radiation. And so it's found that a long time ago now, these animals can survive up to, what was it, like 1,000 times more intense radiation than humans.
Yep.
And so the question is, yeah, it's like, well, why? What about them is being, and it seems so basic of a question. I don't know, to me, Danny, right? Of like, why on earth?
Yeah, I'd heard before some people imply that it's because their bodies are just incredibly resistant to being harmed by it in the first place. But here, what these researchers found is that no, they're actively being harmed by the radiation. It's just that they're able to basically heal themselves as they go without dying. So it's not like they're immune from the harm in the first place. It's just that they've got some sort of a self-repair mechanism, which is really cool.
Yeah, and there was a paper, oh God, I think it was 2017 that was actually getting to this point. And because the question was, where did these genes evolve with? And this study still doesn't answer that, but at least it gives us some of the underlying genetics. And that paper suggested horizontal gene transfer. So genes were jumping from one organism into the tardigrade, and that almost was imbuing the tardigrade with the ability of whatever that gene had. Like, it felt like it was like, I'm acquiring these genes for better survival. And it actually was a very clean-looking paper. Lita actually presented it for a journal club in graduate school, and people loved it. And 2 years later, I think Lita's told this story. It was turned out it was
you told the story last week.
Yeah, contamination.
Funny.
And so it was. This is why when I read this paper, I'm like, I wonder, like, do we need to take any of this with a grain of salt? But I thought it was a very cleanly done experiment set. So essentially, chat, they went identified what gene is differentially expressed, so what gene is turned up to repair the DNA in these animals when there is this DNA damage that's initiated. And Charlie's Dragons, they're good at surviving in normal state as well as in the dehydration state. So there is this ton state, it's called. When the animals are dehydrated, they shrivel up, and actually their DNA breaks when they're dehydrated. And they change their cellular composition to, instead of being water, it's almost like this jelly matrix, so that when they get rehydrated, the cells don't pop. And in this jelly matrix, there's actually a bunch of DNA repair enzymes as well that we think are different from the ones presented in this paper. And I think what's really cool is, if we just get straight to the figures, they did a bunch of imaging. And so this staining here, so the DAPI in pink is DNA. So these little pink dots are just the nuclei of the cells. And tunnel staining in green marks the ends of DNA for broken pieces of DNA. So it doesn't mark the ends of them as in like the tips of your double helix. Instead, it's at breakpoints.
Interesting. Okay.
And so that's what this is.
A lot of breakpoints there, it looks like.
Yeah, so here, so this is not irradiated. You have a tardigrade that's just happily hanging out, and there's no tunnel signal, as you would predict. You irradiate them, and you get a ton of DNA damage. And this is kind of a sanity check as well. So if you were to zoom in on this image, we're at max zoom here, you're getting both signal. So you're getting the pink signal, and it overlaps with the green signal. So you know then the tunnel staining for the end of the DNA, it should co-localize. It should be marked by both molecules because it's DNA, ends of broken DNA, right? And so you get this feature here where all those pink dots are marked also by green dots. And then this was the really wild part, and I think really nicely shown, is after 24 hours, that green goes away.
That's wild. That's so cool. Like, what an elegant indication of that right there.
Yeah, and it's like,
I love it.
It's quick. Like, that's the other, like, surprising how quick it is, that it just responds to the environment. And it's not like it's a little bit of a decrease. It's near, it looks like a totally different
Night
image.
day, almost.
Yeah.
Yeah.
And so, you know, they do the quantification of this as well. So you have, you can do like the intensity, signal intensity. So you can do it in an unbiased way. You can feed it to a software to do the measurements, and you do the comparison between irradiated and not irradiated, and you see that like there's a big increase in irradiated animals, but then it goes back to no difference in irradiated and non-irradiated after 24 hours. So what we see in the image is what you get from the data. And this is one of those things that I really like in scientific papers, where if you can see it, like I want the bar graph too. Like, I'm a purist on that, but I want to be able to see what you're telling me is true. And I think this is one of those nice examples where you can see the data in the image and it presents the exact same thing in the bar graph. Like, it's a very nice, easy to interpret thing. And then it's just different intensities of radiation between the panels D and E. And so it's like, all right, that's really cool. What exactly is the reason for this happening? And so I think, Danny, we talked about this last week, of volcano plots, of looking at different gene expression, right? Of like, what are the differences? So Miss Yvette, folks are wanting to figure out why tardigrades are able to survive radiation. Like, what is the genetic basis of them surviving well? If you basically put them under like a uranium bomb, why are they surviving and humans don't? And so what they get is this here is that volcano plot, which our friend Volcano Doc actually is not connected to. It just, apparently this looks like a volcano erupting. What you want to look at is if you are going under 0, it's the whole plot is a comparison, chat. You're comparing your normal control state to your experimental, and you're measuring differences in genes. If it's under 0, it's genes that go down in the experimental, so you have less of those transcripts, less of those proteins being made. And if it goes above 0, you have more of those RNA molecules made, which leads to more protein. And then there's another threshold, so you can go on the x-axis to either go low or high, and then on the y is your significance intensity. So the higher you go on the y-axis, the more statistically significant what you're showing is. So even if, let's say, you find a gene that's all the way out here and it's at the very, very end of it, it might not be statistically significant. It might just be a very noisy gene. And so you want something that is moving away from 0 but is also high on the scale. And so they actually pull out all of these genes here and they do a little bit of a zoom in, and they find it's all these DNA repair genes that have come into play. Everything from base excision repair, so this is, DNA is made up of bases, where they remove certain base molecules and repair them. So they add in a different base, and it's, if something's mutated, they put in the correct thing, so you find a bunch of genes doing that. You find other DNA pathways, and then you also find the non-homologous end joining is fixing what we saw in this figure with the green dots. So the genes come in and they rejoin the green dots to remake that DNA strand. And so it's actually really elegant, right? Like, you see this image, there's breaking of DNA from the irradiation, it gets repaired, and then the genes that you pull out are things that are repairing the DNA. Like, it kind of,
so cool.
Yeah, it's like a really nice, like logical step by step, push here, which I thought was really neat and really clear cut. They then actually did antibody stains, which, Danny, we talked about, gosh, maybe like a month or two ago now, where you can visually mark a protein of interest. And so here they had that list of genes, right? And from that list of genes, you want to mark a protein of interest. And here that protein of interest may have been this LIG1 DNA repair molecule or PARP3. And what's really cool is that there's, again, it's backing up the sequencing data. So this is at protein level, so they're not marking the RNA, they're marking the protein. And it corresponds. So like in irradiated, there's now high levels of this gene product and not high levels in the non-irradiated. And it's co-localizing with big splotches of the DNA located in pink. And so again, the reason I like this paper is just because it's so elegant. Like you have the data that back, like the bar graphs that then back up the visuals and vice versa. Like I thought it was like, oh, this is really cool to be able to pull these things out.
And I love that the elegance of the method there. It's plain to see like what's going on. Like the DNA is being repaired. Like it's not like this is several layers deep or something, and there it is. You could basically see that in the images. That's really, really cool.
Like, and it's such a strong effect that I feel like you do not need the training to be able to tell. And I think, it's great to have the training, but that to me is an even more striking phenotype. If I don't have to know what this gene is, if I can just look at it and be like, okay, it's not here and it is there, right? That's a very nice thing to work with. Usually it's not that clean.
That's pretty awesome. Yeah. And not only is this elegant and clean and just beautifully illustrative like that, but it sounds like another team independently had pretty much the same results.
Yeah. Let me pull that up.
Beautiful to see that,
And rare, too, that they then even corresponded. So, yeah, there was a second paper. I think this crew was in France. This was a crew in France. I bet there's some shade going on here, Danny, between the two groups because of where these papers are published. So,
eLife versus was it about communications biology?
Let's see what it was. Was it current biology?
Current biology. Sorry, yeah.
So current bio is like a good middle of the road, like really good middle of the road, like not like in the 50th percentile, but like 75th maybe.
So that'd be higher than something like PeerJ or something.
Yeah. There's like, and it has a lot of good historical data to back that up too.
Would it be about the same as like? Do you know the journal Historical Biology?
I do not.
Would it be about the same there? Or?
I don't know that one.
Yeah. Or maybe Proc B or something.
A little bit higher than Proc B. I know like the one I compare it to is like PLOS Genetics. Like that tier of journal. eLife is a weird one because they used to be, for chat coming in, it used to be very prestigious. It was formed by folks from the Howard Hughes Medical Institute, and it was supposed to be this really rigorous set of like a really good new journal. We actually published in it when it was relatively new under the guise of what it was promised to be, and then maybe last year they did away with peer review.
Yeah, that was a weird move to do.
And the logic behind when you submit it, it goes live, and it says
it's almost like post publication peer review. I think is their term of art for it, isn't it?
Yeah.
Something like that.
Yep. And it's like an active it's meant to be an active process and an active review process, and when you click on it, you get to see like at what stage. So like here it says reviewed preprint. But so you can see it above Danny chat. It says reviewed preprint, but that doesn't mean it's actually done. like, and you know, no paper is ever really
It's a work in
done.
progress.
Yeah, I did say all publications are technically should be considered as living documents, but this is a little bit more of that in between space of you don't really know how much of a living document it is right now.
Yeah.
But
Yeah.
all that aside, they did do very similar findings. So here, they just did different stains. So host is the exact same thing as Dappy. It's just a German version of it. You know, it's
really?
the molecule is slightly different, but it does the exact same thing. And then this phospho H2AX is a marker of this, like essentially a marker of DNA damage and of breaking. And Dr. A says eLife right now is above current bio in SJR. eLife is 16th and current bio is 24th in genetics.
Really? Huh.
That's interesting. On the pulse of where people view that I've talked to, maybe I'm just talking to a biased group of folks. A lot of folks seem to be jumping ship from eLife right now as it's transitioning from that review state. Some of those metrics are still historical right now, like before that change. So I'd be curious to see in like 3 years, you know, does it hold where it had been? Do people go back to it, or what's exactly the outcome there? Just because some of those metrics are a little bit still on the older side. But yeah, they do a little bit more here. They do what's called Western blot staining. So they're staining. They not only show you visually the protein that's marking the DNA damage here in these images, but they quantify the protein on this gel. And so you're seeing with increased concentration of the radiation, you're seeing increased amounts of the protein to a certain point, and then beyond a certain point, it's just killing the organism. So they do look at like the extreme side of things. And then yeah, there's the same volcano plot. And actually this plot here, like the zoom in looks almost identical, right? Like you look at that one.
The one on the left does too, yeah.
Yeah, and compare it to oh, let's see, where are my figures? This one here. Volcano Doc, your ears must have been ringing. We were just talking about you. Welcome in. Guys, go follow Volcano Doc. We actually just gave you the fancy shout out, Miss Dr. Professor of Sick Volcanoes. The name is, yeah, this is a volcano plot. I like Dr. A said, it looked a little bit more like it's something crashing inward, like an asteroid coming in, and you have the particulates flying out.
Called a crater plot or something.
Yeah. But yeah, it's called a volcano plot to measure gene distribution between your control and your experimental. And so everything like below 0 is a decreased amount of genetic product, and above 0 is increased amount. And what was interesting was, and I like that they pointed this out in the paper, so they validated the RNA levels with protein. The reason that's important is just because a gene product decreases does not mean that the protein will also go the same direction. So I've had studies where we found the RNA levels go up or down, but the protein levels, there's not that corresponding change. And so it was really cool that they showed that the protein change is the same as the RNA levels. Who's the visitor we're going to have? Which of the pies are there?
Oh, Sweetie Pie. I'm trying to get her to come up on the desk. Supposed to be giving her more of these dental treats for her teeth after a trip to the vet. So, come here, Sweetie Pie. Hey. Come here. She's being shy right now. But maybe she'll jump up here.
Socks is being shy too. She's just curled up in a ball and napping. She's usually on the screen when you're here and wanting to chat.
Yeah. Used to hearing her meow. Yeah.
But yeah, and I thought it... Oh, there we go.
Here she is. There's Sweetie Pie. Yeah.
Oh, y'all have such a cute collection of cats.
Oh, thanks. Yeah, they're something. Like Gretchen. Yeah. Yeah.
So yeah, I thought it was cool that they showed this kind of breakdown as well of the anatomy of the tardigrade.
That's nice to see. Like often in a lot of papers like this, you don't have like a representation of the actual organism.
Yeah.
So I appreciate that as somebody who works on, on critters. It's nice to see that diagram. Yeah.
Yeah, I'm I also don't work with tardigrades, right? Like beyond us looking at during microscopy, and I don't know like what necessarily they're aiming to look at or what they want to look at and what they want to highlight. And so it's nice to have that extra level of detail. Tatum,
Yeah,
sleep
for
well.
sure.
Thank you for hanging with us, Tatum. Reiko, hi, welcome in. So yeah, it was. I don't know. I thought it was really cool to be able to see that level of detail, and then, they also do this additional piece of data. So they do. It's high levels of certain DNA are sufficient to improve bacterial survival after ionizing radiation, and I think this is like the moneymaker here in terms of experiment because, right, if you see these genes are going up from radiation,
Yep.
okay, sure, and you see the proteins are going up after radiation, sure, but never once does that prove that those are the things that are being protective. And so what's really cool is they took tardigrade genes, put them into bacteria, and irradiated those bacteria, and they survive better. Right, and so
That's pretty awesome.
yeah, like that's
Yeah.
to me
Yeah.
that's the really like
That's the clincher.
yeah. The only other thing I would have liked to have seen is the opposite. So this shows that it is sufficient. So it's always back to this phrase in genetics: is it necessary and sufficient? And so necessity is usually indicated by getting rid of the gene in the organism that you're looking in. So if they would have turned off these genes in the tardigrade, and then they no longer survive radiation, that is necessity. And then sufficiency is here, where you're putting it into an organism that doesn't have it. You turn up this gene, and now they survive the stimulus better. Those would have been the two good elements, like to have both. But I think it's really cool that, bacterial survival after this massive amount of radiation, and here you've got the empty vector. That's what you're comparing against. So there you insert a piece of genetic information that has nothing. So that's your baseline that you're comparing against. Yep.
Okay.
And so this is the
Like
most
a control.
important. Yeah, this is your control here. And note that insertion of some of these elements actually destroys survival. And that's their negative control. So these are things that they knew if you turn off will hurt the bacteria, like even more so. And then here is your control that's giving the same. It's just you're putting in an empty genetic piece, and then all of the rest of these are some kind of tardigrade gene, and it goes up over time. And I think that's really really cool. And it's the same thing in the eLife study as well, where they put in these elements. They've got a little bit, pretty figures, and they have the same kind of like tracking. They have some interaction here that are showing a little bit cleaner, I think. So it's more kinds of imaging techniques to show the overlap. Like they actually had better access to higher level microscopes, so you can do the overlay. Like that's just a common way of suggesting these two interact, these two molecules. If you have what is called a frap, so if you have these two proteins, and if they're close enough, they'll excite one another and they'll glow a different color, versus if they're not far apart, they'll glow as two independent colors. And so they had that genetic trick using these tardigrades. You need a super resolution microscope to look, and that further showed it. And then here, yeah, they did human cells. So honestly, like, what's better, human or bacterial? I think it's probably more media worthy, probably, if you do human cells, just because
it's a human cell, yeah, for sure.
Just because you're like, oh, that's cooler. But I mean, I don't think that this means in any way we can inject ourselves with tardigrade DNA and survive a nuclear blast, any more so in a bacterial cell, But it was still cool that there is like, they have this increased level of repair. These human cells are able to utilize the tardigrade gene and repair the DNA. They didn't measure like how close to the original DNA you were, like how good is the repair. Instead, it's just there's less of these dots, so the repair has worked in some way. But it's still really cool to be able to have that. And I think that's it for the figures on this. Yeah, both together, I think it was really nice. They came out at the same time. They have the same conclusion in essentially the same ways too.
Uh-huh. I was surprised at how similar those studies were, and I don't know what the story is there. That's is it just pure coincidence, or um, I wonder what's going on there. Yeah.
So
Yeah.
the approach that they took is very, very standard in terms of like how you'd
Gotcha.
approach the genetic question. Like, right,
Uh
if
huh.
if you have the sequencing stuff like good to go, it's like I see this phenotype. Let me compare these two things by sequencing both. You do the
Yeah.
volcano plot. If you're lucky,
Hmm.
you get candidates. And here, I mean, these candidates were just. I mean, these are really lucky candidates, right? Like you're if your hypothesis is DNA repair, and you
Uh
get
huh.
a huge cluster for base excision and then rejoining the strands and then a bunch of
Hmm.
other like it's essentially leading like yes this is it, and then you just pick the really highly ranked ones and test them.
Uh huh.
And yeah, the good so the reason they tested these genes too is because they knew that the antibodies would cross react, meaning they didn't need to make brand new reagents to work with a tardigrade specific protein. The protein exists in other animals, and so you already have antibodies generated, raised in,
Okay.
like from a mouse that reacts with the
Uh huh.
tardigrade one because they're so close in terms of molecular structure. And so that
Hmm.
makes things a lot easier. If they needed to generate their own genetic tools, you definitely wouldn't have gotten so many types of images because
Hmm.
it's like when we have to make something for an ant, let's say, you have to find that gene of interest. You have to
Hmm.
make a ton of copies of just that genetic element. You have to clone it into a bacteria that'll produce a
Uh
ton
huh.
of that protein product. You then have to take the protein product, send it to a company to inject it into a bunny rabbit or another mammal
Hmm.
for it to have an immune response against, right? You then have to harvest the antibodies from the animal,
Uh
purify
huh.
it, do a re-injection, purify the second batch, and see with genetic manipulation if when you test it, if you get rid of that gene, will you get rid of the band? So it's like there's so
Hmm.
much quality control testing. That process that I summarized in 30 seconds can take 6 months to a year.
Uh
And now imagine
huh.
doing it with all of these.
Uh huh.
So I think that's also why they picked those genes and why between the
That makes
two,
a lot of sense.
yeah, like why between the two papers you're also picking the same genes. And then the fact that
Uh,
it's at the same time, I wonder if saw someone presenting at a conference.
yeah. It's exactly what I was thinking. Like they both saw the same abstract in the abstract volume, or they were attending the same talk, or yeah,
And they were like, "Yeah,
yeah.
let's just get it out in front of us, each other, and just try to both
Hmm.
of us do some kind of front foot kind of thing." But I don't know, Danny. Like,
Right.
what did you
Yeah.
think? Like, not coming from the did the genetic and was there? Did you find like a good logical flow? Are you satisfied with the answer of the way they repair
Yeah.
their DNA are these genes?
That makes a lot of sense to me. I mean, a lot of this stuff is going to sail over my head because I'm not a specialist in this field. But uh, yeah, it seemed like it was well organized, and their results seemed to match their conclusions to the extent that I can tell that as a paleontologist. It was an elegant little paper. I like this a lot. This is pretty cool.
Yeah.
Yeah.
Yeah, thank you for letting me share this one. Again, I think tardigrades are
Yeah.
just fun. I think there's I mean
They really are.
JD mentioned it's in Star Trek too. It's like in Discovery, it's the spore drive is driven by a tardigrade in the new series, right? So there's like in pop culture they're everywhere, and I think it's people's
Yeah.
imagination gets really grabbed by them. But we have a culture of them we look at under a microscope too, and they're cool little creatures. They're like Chad. If you go outside, if you get some lichen or moss, you'll probably find some in your backyard.
That's what I call moss piglets. Yeah.
Yeah,
Shoot, when I was a kid in the '90s, I feel like nobody knew what tardigrades were. Water bears. My first introduction to them was from an old Animal Planet TV show called The Most Extreme, and it was like most extreme survivors. And tardigrade was, they called them water bears in that, but that was number one. And like, I thought they were really cool. And but I feel like nobody really knew about them until recently. They've got some excellent PR. They must have a really really good agent. It's making sure they show up in movies, TV documentaries, and all kinds of stuff. I think I even saw them in one of those superhero movies too.
Yeah, and I think in the Ant-Man movie, because they get shrunken down, there's like giant tardigrades floating around now too. Like they're just all over, and it's not like, I guess, Jurassic Park, where they didn't really imply any science, right? It's not like they're getting stuff wrong. It's just like, here is this creature. Everyone knows it survives well in these extreme conditions, and that's it for the commentary. Versus, like, I love, Danny, when you broke down the latest Jurassic World movie. I loved it so much because, like, right, there was that one with feathers, but it jumped into ice, and like none of it made sense. You're like, what? Like, you have the dinos, you have the science that is cool. You can follow it, and here, at least with the tardigrades and like their PR, no one really goes above of what they can do, and I think it's because they are just, like, from an imagination perspective, so cool in what they can survive in.
Yep, and they look neat, and like they've got this beautiful aesthetic where it's a nice mixture of like wild, off the wall, almost a science fiction-y kind of thing, where they've got the 8 limbs, and the little claws, and the little newt mouth thing.
Yeah.
But they're also cute at the same time. They walk a fine line there, and they're very good at that. Tardigrades. They're cool looking little beasties, and so I don't think there's a lot of incentive to gild the lily there to try and improve on them.
There are a few that are very Nightmare Fuel-like, I find. With the scanning electron microscope images, they just are more teethy, right? Yeah. But the more common one, like Golganak has been dropping into the chat, which is Dr. NPHD's emotes, or like ours is that little, like, the newt mouth, like you're saying, and the innards of it, there's teeth, but it's like, oh, that's kind of adorable, and it's got little feet that it uses to walk around on.
Yep.
Yeah, and Nelf, the tardigrades that end up on the moon, that they survive. So, Nelf, like, there's been...
What would they eat? Yeah.
Well, so they just don't. There's been some studies that show that they've been going into a period of stasis for 30 years. And then that's just... I think that was one where they just brought them back out because they just happened to have... that's when they ended the experiment. Right? And it's not that they, like, found that it was a limit of 30 years. Yeah. It's just that's when
nobody knows how long they can live.
Yeah, and so, like, vacuum of space, like that ton state is really remarkable because, again, like, the matrix that they go in to survive breaks up the DNA, and then it puts them in this, like, glycerol-like solution almost, and so when they rehydrate, their cells don't pop, and the DNA gets stitched back together.
Pretty darn cool.
Yeah, so I don't know, Nelf. Like, that's also, I think, a concern. Like, if we go to Mars, if you find tardigrades, like, I'd immediately be like, yeah, I'd be like, that's... Or like Europa, right? Like, these water-based planets that we're checking in on, they're a creature you have to be really careful on.
I feel like NASA and the European Space Agency have done a really good job of trying to clean off their spacecraft before they get launched. Especially the stuff that's actually going to make contact with a celestial body, if it's a planet or a moon of Jupiter or whatever. But yeah, I don't think the Soviets always did such a good job with that, and so there could be contamination in certain places. Yeah. And
you never know, like, other places that are also launching, or even private industry that, launches up into space. Yeah. What governing body, like, there is no uniform governing body for some of these things, and even if it's not contamination, because it might perturb discovery, imagine if we took something to Europa and killed off anything that's there because we dropped in our own thing.
It's just overrun with cane toads or something.
Exactly. But yeah, I just thought it was a really cool little study. I thought it was, elegant in genetics, and it just nicely added up the data. And again, I thought that it was really cool that there was both studies, like both the eLife one and the current bio one, that, I mean, they showed the exact same thing.
Yeah, it's neat stuff. That's
Yeah.
neat stuff. Should we move on to maybe the ichthyosaur paper? I've got probably about 40 minutes here
Okay.
before I got to take off and eat dinner.
Yes, sir. Let's see. This one, right?
Yeah. Interesting stuff here.
I am confused, Danny, on this paper.
Oh, okay.
Please
Cool.
take us through this evidence and the thought, because this is like, I feel like this is important stuff, but the logic has gone over my head.
Gotcha. Here, let me show. I'll pull up an image of some of these ichthyosaurs. Yeah, that'll work. Open image in new tab, and then I'll give you the link. So you can pull it up as a visual aid. But ichthyosaurs are these enormous marine reptiles that lived in the earlier part of the age of dinosaurs. So they look kind of like a gigantic dolphin or something.
Yeah.
And there are many different kinds. Early on, they were huge, like whale-sized. You had all these big ichthyosaurs swimming around in the Triassic seas. They really just kind of take over after the end Permian extinction, and they're a kind of reptile. We don't really know exactly where they belong on the reptile family tree. We don't think they're particularly close to lizards. They might be closer to tuatara or something. I don't really know what the current thinking is. They're just, think of them as giant ocean-going reptiles.
Okay. How do we know?
Truly huge.
How do we know that they're reptiles versus the dinosaurs? And I know you answer this a lot on your stream, but it almost comes back to the argument in my head of like the delineation between the plesiosaur being, not a dinosaur. Like how, with these bones, like how do we know where they fall into with that?
So we don't know exactly where they are on the reptile family tree, but they've got a lot of skeletal characteristics that are definitely reptilian.
Okay.
From the shape of their skull to even like the histology of their bones. They're very much sauropsids. They're very much reptiles.
Okay, so it's just like...
The only other option would be that they're synapsids, like mammal relatives, and they're definitely not that.
Okay. I guess that's why...
They've got the same,
it looks dolphin-y, like you were saying.
Yeah, they've got the same socketed teeth like reptiles do. It's very different from like a mammalian sort of deal there. They've got those like proper thecanod teeth, or something close to that anyway. And yeah, so they're very much reptiles. And we also have some cool fossils of early ichthyosaurs when they're first kind of taking to the ocean. And so they've still got like, lizardy looking hands and stuff before those fully develop into paddles, like these guys later have.
So it's a morphological comparison between what we have now and what we already have in terms of evidence of what is past reptile.
Exactly.
Okay.
Yeah. Now to get back to the paper, there were these weird bones that have turned up across Europe, and they're really big, but nobody really knew what they were for a long time. And it's been thought that maybe these are dinosaur bones, maybe these are the bones of giant amphibians called temnospondyls. But a few of these have tentatively been identified as ichthyosaur bones, and now this team has looked at a few more of these and actually done histo work, cut the bones open, looked at the interior structure under the microscope, and that matches pretty well with ichthyosaurs. So it's more evidence that there were giant ichthyosaurs swimming around ancient Europe in the Triassic period. So it's cool, and that this is like some detective work. There are these really mysterious bones. Nobody was quite sure what they were. Now it seems like they're probably mostly the dentary bones, the lower jaw bones of the ichthyosaurs.
So then, I guess where I was getting hung up on, maybe it's pretty straightforward, is how do we know? Like, because it seemed like the way it was framed in the paper was that there is this ongoing debate on what these bones are, right, and what they're connected to. Raiders, welcome in. Hi, Ninja Gloppy. Welcome on in, Raiders. The School of Sharks is approaching. Ninja Gloppy, how was your stream? What were y'all up to today? Guys, if you don't know Ninja Gloppy is a professional player of Rocket League, Also does photography of space, and recently came back from Japan. Also has done a photography in Africa too. Really beautiful photos that Ninja Gloppy does. Welcome on in, y'all. Guys, go check out our friend Ninja Gloppy. Photo editing for your Japan trip. Very nice. Any dinosaurs, Ninja? Or perhaps dinosaur bones? Or cool animals, even. I know we had some ants and termite mounds last time. In addition, I think, Danny, you were here when Ninja came in once with elephant photos and, like, the really beautiful, like, high-res images. Monkeys and dinosaurs. Yes, birds. Yes, indeedy.
Nice.
Very cool. And Cherry, nice. Well, Ninja, thank you so much for the raid. Guys, go check out our friend Ninja Gloppy. Also, Misadventures in Astro, welcome in. For those coming on in, y'all, my name is Balint, 1/3 of the Science Stream crew. The other 2/3 are my wife, Lita, and our daughter, baby Alona. Lita and I have our PhDs in molecular biology, doing live science content here on Twitch 7 days a week. We're joined today by Professor Danny Anduza, single greatest science communicator, not just on Twitch, not just on YouTube's, but the entirety of the internet, without whom there would be no science and tech category on this platform. See, he's backing away because he knows it's true. And we're talking fossil science. We talked tardigrade genetics tonight, and we're talking about this really cool study that had me confused, and maybe it's not confusing, y'all, on ichthyosaurs, which here is this really cool image of what these animals look like. I love how Danny said they look like giant dolphins, almost, is the image that I'm feeling.
But they're reptiles, yeah.
Yeah.
Not related to dolphins at all.
And we pointed that out. Danny mentioned, like, you know, here are the possibilities of what could it have been, why it's reptilian, not dinosaur, and then why we discounted it as being mammalian too. But there was this whole controversy on what this could have been, and the researchers did this. I mean, it's a phase contrast microscopy. I guess I couldn't piece together the way the paper was framed on why this was such a why no one knew what this was, like why this was such a mystery.
No one had looked. It's as simple as that. No, nobody bothered to cut open the bones and look. It's low-hanging research fruit, you know.
So was it as something as simple as we don't know what this is, and we have all these hypotheses because we haven't looked?
Yes.
Okay.
You're overthinking it.
Okay, so
Yeah.
Because I was just like, why? I'm like, how is this enough? Like, this seems like it could have been standard. Like, how does this answer this question?
Yep.
And so, is it just that there were ichthyosaur bones that have been looked in the past, and they looked at it here, and it just is a clean match?
So with ichthyosaur fossils, there hasn't been a lot of histology done on them, like cutting open the bones and looking at thin sections under the microscope. So what they did is they examined some from Canada, I think, and they had a similar kind of cortical bone structure. The cortical bone is the bone around the exterior of the bone. So it's like generally pretty tight weave. And it looked really, really similar to these ones from Europe. So yeah, that was kind of the clincher. It's a simple paper.
Okay. I was like, surely I'm missing something really big here.
Nope.
Because the way it's presented is the controversy seems like pretty potent. Like, it's a game changer almost in terms of like the range of these animals.
I don't know if I call it a game changer, but it's that's funny. I'm trying to open up the paper and it keep getting errors here. Finally, it's going. Yeah, it's the sort of thing where like nobody's just bothered to cut the stuff open before. This is a continual theme within vertebrate paleontology, especially with regard to dinosaurs and other Mesozoic reptiles. Is like sometimes you just gotta cut the bones open to see what's going on inside, and there can be a lot of reluctance to do that, even though it could be such a powerful tool for actually figuring out, you know, whatever biological mystery is current.
So what's driving that reluctance? Like, because when we've talked in the past about this, it's like maybe there's one holotype, and it's in a different country, and that's
Uh-huh.
like there's this, like it's old or something in terms of its discovery, and they just worry about damage. Is that the same principle that's
Yes.
governing the fear here too?
With some of these bones, they were collected. Not only are they very, very old because they're from the Triassic period, they're over 200 million years old, but also some of these bones were first collected like 100 or 200 years ago,
Mm-hmm.
and so they're like an important historical part of the collections, and so it could be really difficult to get permission to cut them open. Um, Even though that, that is really important for understanding them. But if I were, say, a collections manager, say I work at a museum, and I'm in charge of the collections, of protecting them and preserving everything, I might be really reluctant to let a researcher saw one of those things open, because that's, destructive sampling. And who knows, maybe 20 years from now, there will be some sort of micro CT scanner that will allow you to do the same kind of a study without actually cutting the specimen open. So, yeah, I don't know, but I guess that's why I wouldn't be a collections manager, because I don't have that attitude.
So
I was trained under the idea that, like, no, sometimes you gotta cut these things open to figure them out, and you can just go out and collect more later. Like, what's important is understanding and publishing, like figuring out these mysteries. The fossils, we should take the best care of them that we can, but sometimes to make an omelet, you gotta crack a few eggs.
is my understanding correct that you're not really destroying the specimen? You're just taking almost a biopsy, right, of the bone?
Yeah, in some cases you can just take a plug out.
Yeah.
So, like, you take a core sample. But with these, I think they actually had to take out a section, and then you cut that further up, and then you make the,
Okay.
pound it onto a slide and grind it down. But what you do is when you cut that section out, say it's an inch wide or something, what you do is you take that and you mold it and cast it. So you have a plastic one that matches it, and then you can glue the plastic one back in between and paint it so it looks like nothing ever happened. That's generally what we did at, like, Museum of the Rockies and other institutions.
So there's not like that way. There's not like a visible destruction of the sample, and you're more likely to be able to preserve it and keep it going. And
Yeah.
okay.
You still have the morphology, so, like, the external morphology will still be preserved because you made an exact copy of that section that you took out. But it is, I guess, destructive, or I don't know. Back at Museum of the Rockies, kind of where I was trained, we didn't like to call it destructive sampling. We like to call it enhanced sampling. Sorry, kind of euphemism for it. But the slides that we would create and the chunk that was taken out, you just put that in the drawer next to the specimen when you're done. So in some ways, you almost have an extra specimen there to look at now. It's like you're expanding your opportunities for gathering data, and I think that's really important.
Did you ever do this kind of microscopy or sample prep?
I helped once. I helped with some researchers, like, grinding down, and you really do have to use, like, a big grinder device in order to be able to grind it down to the point where you can pass polarized light through it on a slide. But I've never really. This is outside of my realm of expertise. Like, I don't do studies like this.
Gotcha.
But I've witnessed this, and I've been around people who do that, and I've helped out a little bit with some of the laboratory work.
So you would, I guess, if I'm understanding the process correct, you take a couple of as thin cuts as you can, right, of the plug that you've taken out, let's say, and then in order to make it even thinner, is that you have to saw down in some way so the light can pass through. And I guess the thing that I would find difficult is, how do you know, like, if you've gotten the right section, right? Because, like, in bone, presumably there's different segments as you're going down. So, like, are there good visual cues for letting you know where in the bone strata you are, so that you're not sawing between strata? If that makes sense.
So what you're doing is you're going at it from a perpendicular angle from the so the bone.
Oh, okay.
The bone like this, and you're doing a cross section of it.
Gotcha. That's. Yeah, I was thinking of it overhead, but by cross sectional, you're gonna capture the whole thing in it. You're not gonna have that be an issue. That is
exactly.
Yeah,
so you're looking at it like you're looking down, face down at a tree stump.
Yep.
Where you've got all the rings like that, basically. It's a perfect cross section. That's what we try to do.
Nice. It's a really cool technique, and I really like the images.
Yeah.
I didn't realize that there was so much, like, with the polarized light, like, there's so much that is revealed in worlds that we don't see, right? Like, even in insect wings, that's a thing. Like, because you see insect wings have these polarized light, and that polarized light has information that the animals communicate, right? And I didn't realize and appreciate until reading this paper how much information we're getting from polarized light from bone.
Yeah. It basically helps you kind of distinguish different parts of the bone architecture. The Haversian canals, the, osteoblasts and osteoclasts, the lines of arrested growth, all that stuff becomes more obvious when you pass polarized light through it. So it's not like the colors really correspond to anything in nature one-to-one. It's more like it helps you distinguish different features. At least that's my understanding of it.
It's like this part of the bone usually is this kind of color because of the properties of it, the optics of it, and then
I think it depends because a lot of that stuff will since these are fossilized, you'll have it permineralized with different kinds of minerals, and I would imagine that if you've got an ichthyosaur bone from one geological formation versus another one, you'll have different settings with different minerals that seep down into those bones, and so it might be different colors under polarized light. It might not be consistent across different geological settings, if that makes sense.
Yeah, definitely. It's a really cool technique, and it's a shame of how rare it sounds. Like, again,
it's getting better.
Okay.
It is getting better. So, like, this is really encouraging to see that histology can be used to solve just kind of a basic mystery about the identity of some bones collected a long time ago. That's really cool to see, and I'm kind of an evangelist for doing histo work like this. If there are any other paleontologists watching who are trained at other institutions, they might be rolling their eyes right now, like, oh, you Museum of the Rockies people, you just want to cut everything open. But there's so much information that you can gain by doing that, and I think it's really important to be able to gather those data through enhanced sampling.
Why is there that eye rolling?
I think. Oh boy, that's a long story.
And that'd be opening a can of worms, or it seems interesting. It seems like a good technique to get all this information. Like you said, the reason that they were able to, I don't know, I took this as this big mystery, the way I read the press release in the paper, the intro, but you were saying, it's low-hanging fruit. You take a look. You would have taken a look, you would have gotten this. This isn't it's only a big deal insofar as you just hadn't done it yet.
Yep.
And so that's surprising to me that it's that.
So much of dinosaur paleontology is like this, bullet. It's just nobody's bothered to do something yet because there's so few of us working in this field, and we often have very limited resources. So there are a bunch of several of the projects that I'm working on right now are just stuff that nobody's ever stopped to do, even though it would seem pretty obvious if I were to explain it to you.
So it's not that this tech is necessarily really expensive.
Since the 70s. Yeah.
Okay, or cutting, no pun intended, edge in terms of newness. It's just you just haven't looked.
Yeah. And I think some of the eye rolling from other institutions might be that at Museum of the Rockies, like with Jack Horner, we were pretty gung-ho about trying to figure out which dinosaurs were young and which were old.
Mm-hmm.
And figure out the ontogeny there. So a lot of dinosaur genera or species that people have named might just be juveniles or adults of previously named genera or species. And we've been pretty gung-ho about figuring that out, and not everybody's a big fan of that because nobody wants to have their dinosaur genus or species downgraded, sunk, as we call it.
They want to keep that Nanotyrannus or Nanotyrannus, which I can never get out of my head now.
Yeah. But yeah, Nano Tyrannus is a good example of that. And it's an ongoing debate, you know? Um, yeah. So there's some kind of headwinds that we're facing there. We're like, people don't like their dinosaurs being sunk or synonymized. But yeah, that's a big thing that we do with histology, and that's. I've got a couple of different projects on the back burner right now with that kind of science.
So do you think that this kind of paper will help almost normalize doing something
It's
like this?
another, it's another brick in the castle there that's being built, I guess. And this isn't anything revolutionary, but it's cool that we can use histo to do something super basic like this, like determine, like, oh yeah, this is an ichthyosaur. Because oftentimes histo is used to figure out if an animal was young or old, how fast was it growing, was it mature when it died, that kind of thing. But here, actually using histology to figure out the taxonomy, to figure out what kind of an animal it is, that is almost kind of new. I'm sure it's been done before for a few different groups. This might be the first time for ichthyosaurs, though, for trying to take an isolated mystery bone and say, oh, this is or is not an ichthyosaur. It's exciting that we can use histo for that. I think that's kind of new, at least for ichthyosaurs.
It is really cool. Is it? I guess it's depending though still on having a database, right? That's curated where you've identified the dinosaur in your classic means, doing the histology, characterizing it, and then having a database that someone can go to and check later, right?
Sure, yeah. Even if it might not even be a database, we're not even at that stage yet. It's just, it's probably like, has someone on this team cut open an ichthyosaur before, looked at the bones in a histological
Oh, so it's
sample?
like it's really new.
Oh yeah, in that sense, yeah. And there's just not that many of us. Like, the paleobiology database is pretty new, and that might seem like the most obvious thing in the world to just like have a database with fossil specimens from around the world. But it's like something in the last few years that the PBDB has really become super usable like that, where it's not just riddled with errors and garbage in, garbage out.
That was what I was gonna say, like the usability of it.
Yeah.
We talked about that in one of our first crossovers, is that it's changed over time, and those changes have made it more usable and more impactd.
Absolutely.
And so it's probably
there's been a ton of work that's gone into that. Yeah.
That's probably the same principle then that would be applied here, right? Is you'd have to standardize it so that you could look at these images. Like if I was specializing in ichthyosaurs and you're specialized in tyrannosaurids, and we wanted to compare, I don't know, just the cut sites, right? We'd have to have been doing it in a very similar way to be able to have an appropriate comparison.
Yep, for sure. Yeah, and there's no like database for anything like that I'm aware of.
And so
there's just not that many of us working in the field right now.
Is it fair to assume that this isn't it's not fleshed out enough to use this as a means to identify new species yet?
Oh, goodness no. You right here they're saying it is an ichthyosaur because all of the genera or species of ichthyosaur that have been looked at, they generally have like a similar kind of bone. You kind of know it when you see it, but like you're not going to be able to identify these down to the species level based on their bone histology, I don't think.
Okay, so all we have from this is ichthyosaur.
Yeah.
Gotcha.
And this could very well be a new species or new genus too.
But we'd have no way of knowing from this.
Yeah. So it's good for saying yes, ichthyosaur, but what species of ichthyosaur? Like, we're nowhere close to that level of specificity with this kind of a method. Yeah.
That's too bad, but I guess it's almost an aim of where to work towards.
Maybe eventually, yeah, it'd be really cool if we could do that, but yeah, it also, I don't know how necessary it would be, because like half the time if you find an ichthyosaur, it might be a new species anyway. So like if you looking at the histo on that and saying, oh, it does belong to this species, like, it's going to steer you wrong. Just because the fossil record is often not that complete, where So I don't know if we'll ever get to that stage where we can identify the species just based on histo. That would require us to have thousands and thousands of specimens cataloged and super well studied, and to have like a much more complete understanding of ichthyosaurs. And we're nowhere close to that right now. We're still at the stage where we're like discovering new genera and species all the time, and this is still the beginning, I guess.
I like your mindset of this, because I think there's two schools of thoughts, at least in the molecular bio field too. It's like there's this new technique, let's just use it on everything, right? Regardless of if it's a worthwhile kind of thing, or like how you're taking with this. Well, we don't need it here. This is like a neat addition. It's a neat tool for some kind of questions, but you don't have to do it on everything.
Yeah.
And so that's I'm just like hearing like the single cell RNA sequencing debate kind of going off. Welcome in, Rosie, from what you're saying, because in single cell RNA sequencing, that was the same issue of, well, there's this new tech, it's really expensive, but it answers a lot of questions. Let's give it a shot for the sake of giving it a shot.
I think a big part of that might just be that you have so many more geneticists working than paleontologists. Where she like, would you guys say the Genetic Society of America? How many members do you have?
That
there?
One's like 5,000.
Okay. Globally, for vertebrate paleontology, we've got like maybe between, I want to say it's like 2,400, it might be closer to 3,000 vertebrate paleontologists basically in the whole world. And we use all different, paleontologists use all different kinds of methods. So it's a much smaller discipline than the genetics, and I think, we don't have, I don't know if we have big fads like that, or like, oh, this new method came out, everybody's using it. Just because, what do you mean everybody? Like, who is we don't have that many people, Yeah.
Totally fair. Yeah, I mean, with the GSA, it's a smaller organization, relatively speaking. Right, like 5,000 is a small group versus like association.
So it's not like every geneticist in the country is part of. So pretty much every vertebrate paleontologist in the world is a member of the Society of Vertebrate Paleontology, plus graduate students, plus undergrads, plus you've got all kinds of enthusiasts and people too who are members of SVP.
Wow.
Yeah, it is the biggest group in the world for. That actually might be an overestimate of the number of vertebrate paleontologists in the world. There might be fewer than 3,000 of us globally. Because it's
Wow. Yeah, I mean, the GSA number that we just cited, that's like the graduate students, postdocs, undergrads, professors, high school teachers, some, like it's a smaller person, but yeah, that's overall a very small number. Like Society for Developmental Biology, I think, is like tens of thousands. That's just developmental biology.
Yeah. Shoot. So.
Yeah. All right. Yeah. I didn't realize again that again makes more sense of why this is a little bit more rare of a technique that we've been chatting about.
Yep.
But it's really cool. Like, I really like the imaging of this. I was a little bit surprised it was. But only phase contrast, because the way it was set up in the press release of like. It almost sounded like it's a revolutionary new type of microscopy.
No, it's not. Or at least that's not the impression that I got. Like this is fairly standard, I think. Looking at the abstract here. I don't think anything about the method was particularly groundbreaking at all. It seems pretty standard.
That's what it sounded like. Like, when I read the paper, that's what it sounded like. But when I read the press release, it felt like it was the way it was written was that it was an extremely new methodology.
That sounds like they're exaggerating that.
Gotcha.
I don't know if I read the press release.
Yeah, it was nice to see though, like how much knowledge that they know about the different bone structures as well. That like on these images, you were able to point out different regions as well. Like it's already known of like, this is a depth of the cortical bone. This is what it's supposed to look like. There's a comparison for it. This is the phase contrast and like the bright light view. Like it was I like we said I love imaging papers, and so it was a really, I think, clean looking image series
Yeah.
that they did here.
I I also really like that. Man, there's like an idea that I'm enthusiastic about, and I deep down I want it to be true. Um, that this paper might help with. You know, everybody always goes on and on about the blue whale and how it's the biggest animal that ever lived and everything. But we've got a few tantalizing hints of ichthyosaurs that might rival the blue whale in size. Um, and because there's just these giant mystery bones from across Europe from the Triassic period, and if some more of these are able to be sections, so you can do a histoanalysis, and it turns out they're ichthyosaurs. Um, it could kind of add evidence to this idea that ichthyosaurs got really, really, really big and may have actually grown bigger than the blue whale. So I don't know. I would just really like to be able to see that happen. Like, shoot, there were, reptiles swimming around in the oceans bigger than blue whales during the Triassic period. That's an idea that kind of captures my imagination. And this method might be used to help demonstrate that in the future.
And for that, would it just be a confirmation that it's an ichthyosaur? Or you'd also take into account, like you said with the cross sections, can this method get you age of the animal too?
It could, yeah. So it could. I think so, at least. I don't know. Ichthyosaurs might have a certain degree of pachystosis, so like they might have thickening of their bones to help keep them from buoying up to the surface. Like as a buoyancy thing, they might have extra thick bones, kind of like I don't know, penguins do.
Mm-hmm.
I'm not totally sure. I don't know enough about ichthyosaurs to say yes or no to that. So if they do have that pachystosis, that might kind of mess up the lines in the bone. They might have some interesting remodeling that would destroy some of those lines of arrested growth, make it difficult to age them. But if it turns out that some of these like super gigantic mystery bones turn out to be from ichthyosaurs, it could point to like there being super gigantic ichthyosaurs. Yeah.
And any idea of what they would have eaten with something that big? Like what kind of predator?
Yeah.
Presumably a meat eater.
Oh yeah. So they were definitely carnivores, like most aquatic animals. But there's been some suggestion that some of these guys were filter feeders, which is I don't quite see how they would manage that. I'm more partial to the idea that these guys were eating cephalopods, so they're eating giant ammonites, they're eating giant squid-like animals, nautiloids, things like that. And you could probably make a really decent living, eating giant squid-like critters. That's what sperm whale do today, and sperm whale are some of the largest of the whales. They're the largest toothed whales in the world, certainly. And yeah. But the thing about some of these big cephalopods is like they're hard to fossilize, especially if they don't have a shell. So there could be like all kinds of giant squid-like nautiloids swimming around in the Triassic, and it would be difficult to find any evidence of them because they're soft-bodied.
Yeah, I mean even the belemite, right, which is that squid pin-like structure. You're only getting that one part of it, and I think there's like the one impression of the animal where it's like the soft body was preserved in the stone. It's like the exact right way. Yeah. So do you think, I mean, this is the obvious like trajectory, right? They competed with like Megalodon or something along those lines.
Megalodon's from way, way, way after. So Megalodon was from long after the extinction of the dinosaurs.
So they wouldn't have had that overlap in resource competition then.
No. There would have been some kind of shark-like chondrichthyan fishes at the time, but I don't think they got particularly big in the Triassic. Like, the biggest animals swimming around in the Triassic seem to have been reptiles. The ichthyosaurs were pretty much unchallenged there, it seems.
Wow.
And, which is cool to think about. Last summer, when I was leaving for the field, I stopped in Nevada at Berlin Ichthyosaur State Park, and we saw some of those ichthyosaur fossils. I might be doing that again this year on my way out to the field again.
Nice.
But that's the same kind of animal, Shonosaurus. The picture that I pulled up earlier for you, that was Shonosaurus right there. And these were once thought to be the biggest ichthyosaurs in the world. They're like 40 or 50 feet long. But some of these like giant pieces of bone from Europe, which seem to be ichthyosaurs, might be like double that length.
Oh wow.
Just enormous, which
Okay.
is really cool.
Wow.
Yeah.
And
Yeah.
we don't, just to confirm, we don't know that ichthyosaur that we just looked at, what species it is. We just know ichthyosaur.
Yeah. Yeah.
And
it would not be a good idea to name it just based on that anyway, because it's so fragmentary that like if you found a complete skeleton that's just missing the dentary, you wouldn't know that it's the same animal. So it's not good practice to name something based on just a scrap. Somebody might do it anyway. We'll see.
That's what I was gonna say. It seems like it can be pretty standard, though. Is it?
Yeah.
But you were saying it probably is, though, given the location and how thin the fossil records are. That it may end up
almost certainly gonna be something new. Yeah. Yeah.
But you wouldn't feel sufficient amount of evidence to be able to name it as such.
No, because that I don't know. It could be nice to have a name and be able to point to it, but you can also just give it kind of a nickname. You know, you could call it, if it was from Bristol, you could call it the Bristol ichthyosaur, and people would know what you're talking about.
Mm-hmm.
You know?
And then once you get more evidence, you can end up naming it.
Exactly.
Yeah. I really like
once you get some diagnostic pieces. Yeah.
Your conservative approach is always really nice. Like it's, I like it's, and I know it makes it hard too because like what we talked about and we won't get to it tonight, but the other paper I'd like to get to next week if at all possible of like that transitional state of when in the evolutionary time loop you call something species one versus evolving to the next species and how that ends up being like at what point do you split that into a new one and like it adds that like both sides have this like big layer of complexity.
Definitely. Yeah, there's a lot of gray area, a lot of subjectivity there, really.
Yeah.
Yeah, I know we don't like to talk about subjectivity in science, but it's a spectrum there. Sometimes nature is messy and doesn't like to fit into our neat little boxes.
Yeah.
Yeah. Yeah.
But no, it was a really really cool paper, Danny. I know I got hung up. I was like, why is this such a big deal? Like it seems like so controversial. But I guess I was surprised that you're just like it's not really controversial, just haven't done it yet, and it's a really cool technique that it'd be really exciting to see more of, and I'm looking forward to I hope to have our crossovers going for years. It'd be really cool, if this time next year you and I looked back on this and be like, yeah, there's been 10 papers since that use the same technique, or something like that would be really exciting to be able to see.
Yeah, let's hope so. That'd be that would certainly clear up some mysteries, I think.
Yeah, definitely. So Danny, I know you gotta get going because we're at about that 45-minute mark. Before you go, we have to do our weekly plug of what's coming up, and also the Amazon page that people can support you.
I think that's depleted now.
It's
Did you
gone.
see? I think it's every item on there, unless Ethan put some new ones on.
Oh, we got a few more.
Oh, there's a few new ones. Nice. Okay. He must have just put those on today or last night. Yeah.
Perfect. So guys, Danny, please do the plug. Let's get the party people motivated.
Shoot, yeah, thank you, Bullen. I'm going to be out digging dinosaurs at the end of May through the beginning of July in Wyoming. And last year we actually did a big fundraiser to raise money. That was almost our entire field budget came from this community, the paleontologizing community. So thank you, everybody. This year we've got a few different sources of funding due to some very generous folks. So I didn't have to do a fundraiser, but Ethan's the expedition leader, a friend of mine. He put together this Amazon wish list for different items that we will need in the field to dig up some of these dinosaurs. We've already got half a dozen dinosaurs to excavate, so it's good that we've got more than a month to do that. But it requires a lot of supplies too, and many of these are reusable, so we'll be using them for years into the future. Chances are we'll be able to pass them down to the next generation of paleontologists. So any support that people can give, if you want to make a purchase that will benefit science in a direct, material way, this is a way to do that.
Where's my drink?
And it's been pretty remarkable the number of items that have already been purchased. So, if you'd like, I already bought a label maker so that anybody who wishes to have their name, their username, put onto the actual item, we can do that. So,
I have the
yeah.
email from last week too, the info.
Yeah, dinosaurcampaign@gmail.com.
Yep.
Yeah. Yeah.
As soon as you said it, I found it again. Yeah, we have it on our Discord too, a way to offer that support. And so here again it is. There's the email, and there is the Amazon link, y'all. And it's I mean,
thank you,
you're helping scientists in the field.
And you'll be able to see these. You'll be able to see these items be used live in the field because I'm going to be live streaming as often as I can. I'm going to shoot for almost every day doing a stream. So it's a wonderful opportunity to do some science outreach to, like, instead of just telling you what it's like to dig dinosaurs, I can show you. So yeah, that'll be for basically the whole month of June. And then I'll be working with some of the same people in Utah later with different equipment mostly. With the Utah Geological Survey from mid-July through mid-August in Utah, digging up some other new dinosaurs.
It's one of my favorite parts of your streams, and again, that's why, chat, please go follow Danny. He doesn't like to admit this, but he is a pioneer of science on this platform. He is the reason why many people will be inspired to do science because of accessibility. Like, any kid can now find a dinoologist, right, and ask those questions. Like seriously, chat. Like, how many times when you were young as a kid, you wish you could ask someone these questions? And we were all obsessed with dinosaurs. Munchkin already is into dinosaurs. She has a book that has photos of pictures of them, and that's one of her favorite ones to read now. And she always flips to the part with the dinosaurs, right? And it's like,
starting her off right. Yeah.
Well, I have to show you, Danny. There's a chicken at the beginning, and he's dressed in a dinosaur outfit. And his name is Darwin, and this little girl's like taking him. It's like, actually, you don't have to dress that way because the dino's in you. And she takes him through like Luca and the evolution of dinosaurs and the.
I want to see this book. That sounds awesome. Yeah.
Next week, it's in Munchkin's room, and I definitely will wake her up. But next week, I'll bring it and show you. I forget the title too, but she doesn't read it yet, but I read it to her, and
that's awesome.
You've changed the name of the game, like. Truly, without you, there would be no science here. You've given it so much credibility. I'm here chasing a dream because of you, and I know many other people are too. Or just the amount of knowledge that you've imparted onto a community on this platform is incredible.
I'm happy to be of service. This is what I was trying to do when I joined Twitch, and thank you for taking that dream and running with it, Bullet, because it's pretty awesome, the outreach that you do, 8 or sometimes 9 days a week, you and Lita, it's a welcoming community here, and like trying to demystify science for everybody, make it casual, make it welcoming, make it easily understandable. You're doing excellent work here, and I'm proud to call you my colleague and my friend.
Right back. I have the favorited photo of the two of us finally having that photo together. And we'll have to chat about an upcoming panel as well.
Yes.
I think
I'm coming up with some ideas for that, so we'll discuss that very soon. We should talk on the phone sometime.
Yeah, whenever. Yeah. Just let me know whenever you're up to it, because I know you're gonna be out in the field, and it'd be better for us to get that off your plate before having to go to the field.
Yeah.
But
for sure. Yeah.
Danny, thank you so much. Y'all remember, go follow Danny. Tomorrow's Thursday Burbs Day that y'all have with a lot of birding photos. And then the big other thing coming up on Friday, I forget. What's the next big...
I forget too. What have I got on the calendar? It's loading. It's Velociraptor Awareness Day tomorrow. I forgot. So we're going to be talking all about Velociraptor tomorrow, I guess.
Nice.
Get ready for that. And then nothing special planned for Friday, but we'll have some fossil news. We'll talk about some new dinosaurs just published, that kind of thing. So yeah.
And always audience Q&A.
Yes. That's the perennial thing. That's the bread and butter of our streams. So.
It's amazing. I just love it so much, Danny. Thank you so much for everything you do.
Of course, Belinda. Thanks for a wonderful discussion, and I'm looking forward to the next one.
Yes, sir. Have a great one. We'll see you tomorrow.
You too. I'll see you around. Bye, everybody.
Bye, Danny. All right, y'all. That was the lovely and amazing Professor Danny.